Welcome back to Minty, and we're here with the part 2 of investigating ecosystems. Remember, we're following the CSEC biology syllabus. Let's get straight into the video. So we're going to start with measuring abiotic factors. And as you can recall in the previous videos, abiotic factors are the non-living factors. So we're going to be looking first at the climatic factors. Climatic factors, as the name suggests, relate to the climate or the weather. So temperature refers to how hot or cold something is. We can use an ordinary thermometer or a minimum thermometer. Humidity refers to the amount of moisture in the air. So we normally use a wet and dry bulb thermometer. Light intensity. So the light meter is used by pointing in the direction of the maximum light intensity. Rainfall. Some of you might have had an experiment where you had to use a rain gauge or create a rain gauge to measure the rainfall in one day. So we're now going to look at the edaphic or soil factors. pH. So we sometimes want to look at the pH of soil, rainwater, water in rivers and ponds, and we use a pH probe and meter. Some designs can also measure the amount of moisture in the soil. So usually if we're using a indicator, a universal pH indicator, there's a color scheme that we tend to look at. So red means it's very acidic, purple means very alkalinic or basic, and green means neutral. We can also look at rock particle distribution using the sedimentation test. So we basically shake a fixed volume of soil with excess water in a measuring cylinder, and we leave it to settle and measure the volume of the different types. The largest will sink first. So you can see stones and gravel, they're the heaviest and the largest, so they tend to lay at the bottom of the container. Sand may form on top of it, then clay particles, and at the top we have humus, which is the floating litter. So we can also find the water content and humus content of soil. For water content, first we weigh the soil and we call this weight X. We heat the soil at 100 degrees Celsius, which we know is the boiling point of water, to dryness. We reweigh the dried soil and we call this new weight Y. We find the difference between X and Y and we put this all over X. The difference between X and Y actually represents the weight of the water that was evaporated through drying. But we want to find this weight as a percentage of the weight of the soil before we dried it, so we can know what percentage of water was in the soil. Humus content. So we have the weight of dried soil, which we have here, Y. So we will heat this dried soil to red heat to burn out all the humus. And we can use a Bunsen burner, so we can probably heat it for about 30 minutes, or we can use a lab furnace, heating it around at 400 degrees Celsius. We then will reweigh the soil and we call this Z. And similar to water, we want to find the percentage of humus that was burnt out. So we're going to find the difference of Y and Z, and we put this all over our original weight Y. And we multiply this by 100 to find the percentage of humus. So we can also find the air content and the water holding capacity of soil. In terms of the air content, we want to half fill a measuring cylinder with soil and measure the volume of soil, and we can call that A. We will add water of a greater unknown volume and call that B. We then measure the new volume of soil and water, which we can call C. So the percentage of air will be A plus B minus C, and divide that by A. We expect that soil has air spaces, so when the water is added to the soil, it will fill those air spaces, so the new volume will actually be less than A plus B. So A plus B minus C represents the air that would have been displaced. And we want to find that air as a percentage of the volume of the soil that we began with. We multiply by 100 to find it in percentage. Water holding capacity. So we can place a fixed volume of dry soil in a funnel, like so, with a wet cotton wool. We will then pour a known volume of water through the soil, and we call that R. So say we pour 50 mils of water through the soil. We then measure the water that drains through the funnel into the measuring cylinder, and say this volume was 25. The volume of water that would have been held in the soil would be 50 minus 25. So 25 mils of water would have been held as it passed through the funnel. So the volume of water held can be found by finding the difference of the volume of water before you passed it through the soil with the volume of water that collected in the measuring cylinder. So we can also look at other abiotic factors, such as wind speed, and we can use an anemometer as you can see in this diagram. We can also look at wind direction using a wind vane, where you can see your north, south, east, and west directions. We can find the speed of flow in an aquatic body, such as a river. And we find this by finding the distance traveled by a floating object on the river, for example, and the time taken for that floating object to travel that distance. Because speed is equal to distance over time. Salinity is used to differentiate salt water from fresh water. And lastly, we can look at the direction of a slope. This can actually help us in determining how light can penetrate trees, for example, more directly or more obliquely, and why certain areas may have greater growth of plants than other areas. Don't forget to like the video and ensure that you hit that subscribe button below so that I can continue to make more videos for you.